17 million.
That is a staggering number, isn't it?
It's honestly hard to even visualize, but when you realize what it actually represents, it stops being just a statistic and starts feeling a little, well, overwhelming.
Right.
That is the number of different genetic combinations possible at the exact moment of conception. Two parents, one moment. 17 million distinct ways the biological dice could land.
And that's probably a conservative estimate, really. Before you even factor in the environmental chaos that comes later, it really reframes the whole concept of individuality.
Yeah.
We tend to walk around thinking we were always meant to be exactly this way. But biologically, you're essentially the winner of a massive microscopic lottery.
That is the perfect setup for where we're going today. Welcome back to the Deep Dive. Today, we are stripping things right down to the studs. We're looking at the dawn of development. We've taken a massive stack of research on genetics and heredity, and we're going to try to answer a fundamental question. How do we go from one single invisible cell to the complex, walking, talking human being you are right now?
And to answer that, we have to talk about genetics. At its core, it's just the study of heredity, the mechanism of how parents pass characteristics down to their offspring.
Okay.
It's this fascinating tension where the biological process creates someone who is remarkably similar to the rest of the species. 2 arms, 2 legs.
Right.
But at the same time, genetically completely unique.
So looking at our roadmap for this, it feels like we're reviewing the construction project of a human. We've got the hardware, the biology itself,
Hardware, yeah?
the software, how traits get passed down. Then we've got the system crashes, mutations and disorders. And finally, the user experience, the dance between nature and nurture.
That's a very good way to map it out. We are looking at the code that builds you.
Okay, let's start with the hardware then. Where is this code actually stored? I mean, we all talk about DNA, but where does it live?
So for that, we have to go to the headquarters, the nucleus of the cell. Inside the nucleus of every single cell in your body, you have chromosomes. Now, different species have different numbers. Fruit flies have fewer, dogs have more. But for humans, the magic number is 46. 23 pairs.
Half from mom, half from dad.
Correct. And geneticists make a distinction here that's really important. The first 22 pairs are called autosomes. They handle the general construction of the body.
Hair texture, eye shape, that kind of thing.
Exactly. But the 23rd pair, those are the sex chromosomes. They have a very specific job regarding biological sex. We'll get to that.
Okay, so we have the chromosomes. But if we zoom in, like with an electron microscope, what are we actually looking at?
You're looking at genes. Chromosomes are essentially just tightly packed bundles of genes. The gene is the basic agent of heredity. It's a chemical molecule.
And if you unravel that,
That's where you find the famous DNA, deoxyribonucleic acid.
the double helix, the twisted ladder.
Exactly, that iconic structure. And that structure isn't just for show. DNA basically has 2 jobs, and I think most people only ever focus on the first one.
Which is making babies.
Right.
Inheritance.
Right. Its first job is to pass genetic material to the next generation. But second, and this is the part people forget, it provides the daily instructions for your cells to make proteins.
Oh, interesting.
It's not just a blueprint for the baby, it's the operating manual for the adult. Every enzyme, every hormone, every structural part of your cells is built because DNA told it to be built.
So it's the architect drafting the plans, but it's also the foreman shouting orders at the construction site every single day.
That is a great analogy. It's directing the daily function of all your systems. Now, there's a concept here that the research highlights, and it can be a stumbling block. It's the difference between what's written in your code and what actually looks back at you in the mirror.
This is genotype versus phenotype. I always had to stop and think, which is which?
Okay, think of it this way. Your genotype is your total genetic makeup. It's the full data file locked in a safe. Everything you inherited, hidden or visible.
And the phenotype is?
Your phenotype is the visible result. It's what happens when those genes interact with each other and, crucially, with the environment.
So genotype is the sheet music, and phenotype is the actual concert.
I like that, yeah. Because you can have the notes on the page, the genotype, but maybe the room is cold or the instrument is tuned differently. That final output is the phenotype. You might carry a gene for a certain trait, but it might never show up in the performance.
Okay, that makes sense. Let's talk about how we get those genes in the first place. The research breaks down 2 cell division processes, mitosis and meiosis. I feel like high school biology rushed this, but the difference seems critical.
It is critical. They have completely different goals. Mitosis is about maintenance. It's copy and paste.
Like a Xerox machine.
Exactly. If you scrape your knee, you need new skin cells. Your body takes a skin cell, duplicates the 46 chromosomes, and splits. You get a daughter cell with the exact same 46 chromosomes. It's a clone.
So mitosis keeps me being me. It's for growth and healing.
Right. Meiosis, on the other hand, is about creation. This process only happens in the gametes, the sperm and the ova. Here, the cells divide differently. They divide twice. And the result is unique because the cells end up with only 23 chromosomes.
They're effectively half shells.
They have to be. They're waiting for a partner. If a sperm and an egg both had 46 chromosomes, the baby would have 92, and the system would crash. So meiosis strips the data down to half.
And at conception.
The sperm and ovum join, the two sets of 23 combine, and you're back to 46. That's the moment the 17 million possibilities come into play. It's a massive card shuffle.
And once that happens, mitosis kicks back in to grow that single cell into a human.
You got it.
Now, speaking of that moment, let's settle the playground debate. Who determines the sex of the child? Mom or dad?
Biologically speaking, it's entirely on the father.
Dad holds the cards.
He does. Here's how it works. The mother's ovum always contains an X chromosome. Always. She has two X's, so she can only give an X. But the father is XY. His sperm is a toss-up. Some carry an X, some carry a Y.
So it's a race.
Essentially, if an X-carrying sperm meets the egg, you get XXA biological female. If a Y-carrying sperm gets there first, you get XYA biological male.
The source material mentions something interesting about the size difference between those two chromosomes. I hadn't realized how distinct they are.
Oh, they're very different. The X chromosome is significantly larger. It carries a lot more what we call genetically rich information. The Y chromosome is, it's a bit lighter on data, much smaller.
So in terms of raw data storage, the X is the heavy lifter.
Definitely.
Okay, so we've made the human determine the sex. Now we have to figure out what traits they actually get. This brings us to the rules of inheritance. This feels like where the math gets complicated.
It's basically a matching game. You have pairs of genes, one from mom, one from dad. If the version of the gene, the allele, is the same from both parents, you're homozygous. If they're different, you're heterozygous.
And when they're different, that's when they have to fight it out.
That's the classic dominant versus recessive battle. In the standard model, if you have one dominant allele and one recessive, the dominant one wins. It acts. The recessive one is repressed.
It's like having a loud person and a quiet person in a room. You only hear the loud one.
Exactly. For the quiet one to be heard, for that recessive trait to show up, you need two of them. You need to inherit the recessive gene from both parents.
And this is where it gets really interesting. The research points out that biology doesn't always play by these strict rules. There are rule breakers.
Biology loves exceptions. Life is rarely binary. One fascinating one is incomplete dominance. The classic example is with flowers. You cross a red flower and a white flower, and if the red gene isn't fully dominant, you don't get red, you get pink.
They blend together.
They blend, but then there's codominance. This is where neither trait backs down. They both show up fully. The example in the text is a white cow and a red bull producing a roan calf.
So it's not pink.
No, it has individual red hairs and individual white hairs.
So it's not a smoothie, it's a fruit salad. Everything keeps its own flavor.
That's a good way to picture it. And this complexity matters because most human traits aren't a simple switch. We call those discontinuous variation. You have 5 fingers or 6. It's either...
Wait, hold on. Is 6 fingers a real genetic option?
It is. It's called polydactyly. And oddly enough, we'll get to why that's surprising in a minute. But most traits, intelligence, skin color, height, are polygenic.
Meaning many genes are involved.
Yes. It's the interaction of several genes working together. That's why you see continuous variation. People aren't just tall or short. There's a whole spectrum of height because multiple genetic levers are being pulled.
So usually the system works.
Yeah.
The code is copied, traits passed down, genes win or blend, and we get a healthy human. But sometimes the file gets corrupted.
Yes. We call these mutations. A mutation is a change in the chemical structure of a gene. It can happen because of a typo during cell division or from environmental influences, radiation, chemicals. The code gets scrambled.
Like a typo in a manuscript.
Exactly. Sometimes extra code is added. That's an insertion. Sometimes it's deleted. Or it can be a point mutation where just one single letter of the genetic alphabet is swapped.
And these glitches can lead to genetic disorders. The source material breaks them down into categories, and it really shows how fragile this whole process is.
It is. And distinguishing the mechanism matters. First, you have dominant gene disorders. This is scary because you only need one bad gene to get it. If one parent passes it down, you have it.
What are some examples of that?
Huntington's disease is one, causing degeneration of the nervous system. Achondroplasia, a form of dwarfism, is another. And getting back to the fingers polydactyly, having extra fingers or toes is actually a dominant trait.
That is wild. I always assumed that would be recessive because it's so rare. If it's dominant, shouldn't we see it everywhere?
You'd think so, right? But dominant doesn't mean common. It just means if the gene is present, it shows up. It's rare because very few people have the gene in the general population, not because the gene itself is weak.
That clarifies a lot.
Yeah.
Okay, then you have recessive gene disorders.
Right. For these, you need to inherit the bad gene from both parents. If you only have one, you're just a carrier, usually healthy. This is how things like cystic fibrosis act or sickle cell disease. Tay-Sachs and albinism also fall into this category.
Then we have the sex-linked disorders. These are on that 23rd pair.
Usually the X chromosome. And this is where that size difference matters, because men who are XY only have one X. If there's a problem on that X, they don't have a backup to mask it. Women have two X's, so if one is faulty, the other usually covers for it.
Which explains why things like hemophilia, the blood clotting issue, are so much more common in men.
Exactly. They're flying without a net on that chromosome.
There was one disorder in the list that really caught my eye, testicular feminization syndrome.
Yes, that's a fascinating example of the genotype versus phenotype disconnect. In this case, a person is genetically male. They have XY chromosomes. Their genotype is male. But a mutation means their body doesn't respond to male hormones.
So the signal is sent, but nobody picks up the phone.
Precisely. So despite having XY chromosomes, they develop a female phenotype. They look biologically female. It just drives home that it's not just about what genes you have, but how your body reads them.
And finally, we have chromosomal disorders. This isn't just a typo. This is a whole missing volume of the encyclopedia.
Correct. A missing, duplicate, or damaged chromosome. The most well-known is Down syndrome, trisomy 21. Trisomy means three bodies, three copies of the 21st chromosome instead of two. There's also trisomy 18.
And the research notes a specific risk factor here with age.
Yes, the risk for these increases with the mother's age. The problem usually originates during that tricky meiosis phase. As the ova age, that splitting process becomes more prone to errors.
It's a heavy reality of this lottery. Some disorders are manageable, but others are, well, often fatal in early childhood. It underscores the fragility of it all.
It does, and the support networks for families vary wildly depending on how rare the condition is.
I want to pivot now. We've been talking about genes as if they are destiny, like we were pre-programmed. But the source material introduces a third element that complicates this.
Yes. This is where we move beyond just genes. We enter the world of epigenetics.
It's a buzzword you hear a lot, but I'm not sure everyone really gets what it means.
Okay, think of your DNA as the hardware of a computer. Epigenetics is the software that tells the hardware which programs to run. It doesn't change the code itself. The DNA sequence stays the same. But it changes how the code is read. It turns genes on or off.
So you could have the gene for a trait, but epigenetics basically puts a piece of tape over the switch so it never gets flipped.
Exactly. And this starts early. The research talks about imprinting. This means that for some traits, it actually matters which parent you got the gene from. A gene might be expressed one way if it came from mom, and completely differently if it came from dad.
And the implications here are huge. The research suggests this influences longevity, metabolism, even susceptibility to stress.
Right. And here's where it gets mind-bending. Epigenetic information might be influenced by your environment or even your parents' environment.
So what my grandmother ate could technically tweak how my genes are expressed today.
That's what the preliminary research suggests. It just blurs that line between nature and nurture. It suggests nurture can actually leave a mark on nature that gets passed down.
Speaking of nature versus nurture, the source material is pretty clear that we need to retire that phrase.
It's outdated. It's not a war. It's a dance. We used to ask, is it genes or environment? Now, through things like twin studies, we're learning that it's almost always both.
But there was one concept in the notes that really stopped me in my tracks. The active genotype-environment relationship. This feels like it flips the whole script.
It really does. We usually think environment shapes the child. You put a kid in a library, they become a reader. But this theory suggests the child's genetics shape the environment they choose.
Can you unpack that?
Well, imagine a child who is genetically predisposed to be social and high energy. That's their biology. Because of that biology, that child is going to seek out environments that fit. They'll ask to join the soccer team. They'll run to the playground.
And by being in those environments, they become more social and more active.
Exactly. The genes drove the choice of environment, and the environment reinforced the genes.
And it works the other way, too.
Ideally, take a child who is genetically introspective, quiet. They aren't going to ask for the loud birthday party. They might seek out a library, a chess club, a solo hobby. They curate their own world to match their biology.
That is profound. It suggests we aren't just passive victims of our environment. We're actively building a world that reinforces who we were born to be.
It's a feedback loop. You can't separate the dancer from the dance. The environment shapes you, yes, but your genes help determine which environments you step into in the first place.
That is a lot to process.
It is. We started with 17 million possibilities, the sheer miracle of you existing. We went through the double helix, the gamble of meiosis, the strict rules of inheritance, and the chaos of mutations.
And we landed on the idea that our genes are still quietly steering the ship. They aren't just determining our eye color. They're nudging us towards certain friends, certain hobbies, certain lives.
It connects everything. The microscopic code in the nucleus isn't just a blueprint for your body, it's a blueprint for how you interact with the world.
Which leaves me with a thought I'm going to be chewing on for a while. If our genes subconsciously push us toward environments that reinforce those same genes, how much of our free will in choosing our hobbies, our partners, our lifestyles is actually just our DNA seeking a comfortable home?
That is the ultimate question, isn't it? Are we the driver, or are we just the vehicle for the code?
I think on that existential note, we'll let you stare into the mirror and wonder about your own 46 chromosomes. Thanks for listening to this deep dive. We'll catch you on the next one.